All right. Thank you everyone for joining us tonight for another round of monthly Nashville Data Nerds. Just a couple of quick introductions. First, I'd like to introduce myself. My name is Alex Antonison. Today's sponsor is Antonison Consulting Group, which is my consulting company. Real quick about us is we, work with our clients to provide solutions that foster growth and efficiency in a wide variety of services. Feel free to check out our website if you're interested. Some associated organizations that I cannot say enough about. First being Nashville Software School, being a nonprofit that offers boot camps in various technology areas. And if you're looking on, finding that second career, I couldn't recommend a better organization to check out. Another is Penny University. It's a group of individuals that come together to have conversations about topics of general interest. We are primarily based out of a Slack, and we use that to kind of drive that conversations. And last but absolutely not least is Suneethi here to talk with us about her work on predicting side effects due to interactions between a pair of drugs. Real quick, as we all know, having a group discussion in Zoom can be very challenging. So I will be moderating all the questions. As questions come up, please post them to the Zoom chat. When there's a decent pause, I will pose the question to Suneethi. That'll be for about the next 30 minutes. And that just gives Suneethi enough time to present the topic. The last 15 minutes, we do kind of generally open it up and then kind of have that general group discussion. So without further ado, I'm going to turn it over to Suneethi.
Thank you, Alex, for this opportunity. Let me share my screen. All right, can everyone see my screen? It should read predicting side effects from drugs chemical structure.
Looks good.
Okay. Good evening, everyone. This is my capstone project that I'm presenting. So the goal of the project was to predict the side effects of drugs based on the chemical substructure between two, how they interact, how the two drugs chemical substructure interact. Just a little bit background about myself. So I was a postdoctoral research fellow / analyst. That's what I did mostly as a research fellow, analyzed research data. And then I joined as a data scientist bootcamp at Nashville Software School. And then currently I'm a teaching assistant for the 2020-21 data science cohort at Nashville Software School. So the agenda for today's talk will be to give us a brief introduction of why I wanted to do this project and what are the data sets that I used, and some little bit of data exploration, what I found out with the data exploration, and then what models I used to predict and what are the assumptions or limitations of those using those models. Also, what are the assumptions that I used during pre-processing steps, and then some future directions, and then what are the conclusions, what I learned from this project overall. So the goal of the project again was to predict the side effects of when two drugs interact with each other based on their chemical structure. So we all know that pharmaceutical drugs are a necessary evil. It is necessary because it helps us treat symptoms, but it is evil too because it can result in side effects, which usually or sometimes are mild, sometimes it can be life-threatening. But when two drugs interact with each other, some drugs, they do not like each other, and that can lead to serious or life-threatening side effects. An example is the anticoagulant drug. When it is combined with a non-steroidal anti-inflammatory drug, can result in severe bleeding. So the pharmaceutical drugs are nothing but they are chemical compounds. They're made up of chemicals. And these chemical compounds, they can interact with not only with each other, but with other proteins in the body or with genes in the body causing the side effects. And there are similar studies that are done using different approaches. So the reason, the motivation behind this project was, this was during when the coronavirus pandemic hit US, and all of us were trying to figure out if we can use a specific drug for this infection, and there were contradictory results. Hydroxychloroquine was one of the drugs that was suggested, but it resulted in several side effects, or it didn't seem to be good for treating coronavirus infection. So I wanted to see what happens when two drugs interact. Can we predict really some side effects before it goes into clinical trials? It would be beneficial for pharmaceutical or biotech companies, because usually pharmaceutical or biotech companies, they spend like 10 to 15 years before they can bring drugs into the market. They have to do several clinical trials to see what side effects side effects happen in patients from different health background. And they also spend a lot of money into it. So if we have a model that can predict the side effect even before the drug enters into the clinical trials, it'll be easier, or it'll be easier for pharmaceutical or biotech companies to reduce the amount of time and the effort that they put in bringing all those drugs through the clinical trials, and before they enter into the market. Usually it takes about several drugs screening before one drug get into the market. So all those steps can be minimized. It is not the absolute solution, but it can help in reducing a lot of effort and time that is spent in discovering several drugs and then bringing one to the market. So the data set I used, I would call the first data set is called two sites. It had drug 1, drug 2. Remember, the goal was to see what happens when two drugs interact with each other. So I wanted a drug-drug pair, and what are the side effects caused due to a drug-drug pair. And the two sites had about 42 million observations before pre-processing. And in that, the drug 1 that is in the unique drugs in drug 1 column were 1716 drugs, and drug 2, there were 1842 drugs. And the drug-drug pairs, of course, one drug can repeat in either in drug 1 or drug 2. So the drug-drug pair resulted in like 211,990 observations. And those resulted in unique side effects of 12,726. And the second data set that I used was a chemical structure data set, since that was the features that I wanted to use to see if we can predict the drug side effect. There were 888 drugs with 881 different chemical substructure for each of those drugs. Sorry. So when I did a little bit of data exploration, I found that the most common side effects, the 10 most common side effects were these. Since the data set had, as I mentioned, there were like about 42 million observations with about 1,500 drug pairs, unique drug pairs, and different side effects. I wanted to focus on only the most common side effects, just in the interest of the computing power, and because it's a very long and also a wide data set, and it'll be really hard to run this in a model. So for the sake of simplicity, I wanted to look only at the most common side effects. I also looked at what drug-drug pair caused the highest number of side effects. So I don't know if it's surprising or not, but aspirin with these different types of drugs caused the maximum number of side effects. And the side effects were not really life-threatening, mostly anemia. What stood out is malaria. I'm not sure if this was a cause of the interaction between the drug, or it was just something that happened due to secondary effects. That's something we would not know just looking at this data set. So before I build a model or run a model on this data set, I had to pre-process because it's a wide and also a long data set. So we had the two sites. I had the two sites data set, which had drug 1, drug 2. That's what is shown here. Drug 1, 1716 observations, drug 2 with 1842 observations, and with a total observations of 42 million. There were duplicates, and so I removed those duplicates and selected for the most common side effects, which were nausea, vomiting, diarrhea, difficulty breathing, fever, and pneumonia. I'm actually very surprised. Pneumonia is in the top 6 most common side effects of the drugs. Again, I'm not sure if it's due to the interaction between the drugs or it was some kind of secondary effects. After after removing the duplicates and selecting for the most common side effects, there were total observations of 926,000, which was easier to handle in building a model than without selecting for those. And there were 1,677 drug 1 and 1,805 drug 2 drugs. So these side effects were in the form of names. So this has to be converted into numerical values before I could run a model. So I used Pandas library of get dummies to convert those into either 0 or 1. So for example, this drug, aminocaproic acid with acetaminophen, causes diarrhea. So it will be given 1, dyspnea 1. So that means 1 is present, 0 is absent. So in the second pair, diarrhea is absent. So it is given numerical value of 0. Now I have to also merge this data set with a chemical structure data set. So that we can use the chemical structure as features to predict the side effects, which is diarrhea, dyspnea, and nausea. So now the chemical structure data set has only one drug column. And then it was followed by several chemical structure. Again, those were in the form of 1 or 0. So substructure 1 is present with respect to carotene. 0 means it is absent for GABA. So I did merge in such a way that I merged this data set with this one based on the drug 1. And that resulted in the observation of 126,951 observations with a unique drug of 662. So we would lose a lot of data here because we are merging and we are only taking those drugs that are available, whose chemical structure is available for drug 1. The same I had to do for drug 2. And I got finally 676 drugs. So this is how from merge 1 it looked. So we have drug 1, drug 2, the side effects, and the substructure for drug 1. And then for merge 2, we have drug 2, the side effects, and then the chemical structure profile for drug 2. Now, I have to again merge this in such a way that I had the chemical substructure profile for the drug-drug pair. Now, before I do that, since there are 881 drugs, chemical substructure for each of those drugs, that'll be 1,600 something chemical substructure profile for drug-drug pair. And I had to simplify this again in such a way that there was only one chemical structure profile. This is not the best way to do, but I felt this is the starting point where I can start with something and then change later on. So what I did is when I have the chemical structure profile for drug 1 and drug 2, I took the maximum value of either of between those two. So what I mean is, so this is the chemical structure 1, 2, 3, 4, 5 for drug 1 and 1, 2, 3, 4, 5 for drug 2. So what is the maximum of 1 and 1? That will be 1. And for example, 0 and 0 will be 0. And 1 and 0 will be 1. So it is oversimplification, I agree, but that's something I had to do to build the model. So finally, I had, after merging with the respective drugs and made sure that the chemical structure profile was specifically for the specific drug-drug pair, I got a data set where there was drug 1 of the drug-drug pair, drug 2 of the drug-drug pair, and the side effects of those drug-drug pair and the chemical structure profile of those drug-drug pair. Okay. So just I wanted to give a very, very simple introduction for machine learning model for those who are new to it. So when we do a machine learning, there are 2 different types of models, supervised and unsupervised machine learning. So supervised, unsupervised we're just looking at the pattern, where supervised is you have specific features that, and you are expecting an output based on those features. So the supervised can be further classified into regression and classification. I won't go in detail about regression. So basically regression is you have values instead of 0 or 1, you have like actual values. Whereas in classification, which is the data set that I showed, which is an example of the data set I showed, there will be either 0 or 1. And then you predict whether it is present or absent in the form of 0 or 1. For example, is cancer present or absent? So present is 1, absent is 0. And is email spam or not? So those are the examples of classification models. And I have mentioned a link here which explains in detail about the different classification models. So if you're interested, I recommend reading that. So now we have the processed data set, pre-processed data set. Now I can build a model using that final data set. I use different classification models, which I'll get into later. So when I did an exploration of the data set, after pre-processing, I see that there is an imbalance in the data, which means there is one feature which is represented more than the other. So it will be hard when we build the model, if we have an imbalanced data, the way the algorithm functions is that it'll start, if there are certain features that are in the majority, then the algorithm will select for those majority of the features, which will result in a bias. And that will not be an accurate model to use, and it'll be hard to conclude using an imbalanced data set. The reason that the data set is probably imbalanced is because I selected for the most common side effects, and there are a lot of side effects, those side effects that are very common. So the features that I used are the chemical substructure, which is the 881 different chemical substructure, which are represented as 0 or 1. In order to balance, as I mentioned, it will not be a right way to build a model with an imbalanced data set. So to balance the data set, I used a feature in the sklearn library, which is called oversampling. So basically what it'll do is that whichever is the minority data set, that will be added, like the algorithm will add more of those minority data set in such a way that it is equal to the majority data, majority features. I don't know if I explain it right, but do ask me questions if it's not clear. So finally, the data set I had was the drug 1, drug 2 side effects, which is converted into 1 or 0, 1 meaning present, 0 meaning absent, and then followed by chemical structures. So the chemical structure will be the features that I use to build the model to predict these side effects, whether diarrhea, nausea, or vomiting, or whatever is that. So since it was an imbalance, since it's a classification problem, I used, as I mentioned, I used different classification models, which is a logistic regression is one of the classification models, random forest, lasso, and XGBoost. And the way I did these prediction is that I didn't want to predict all the side effects using the chemical substructure profile. So what I did is I predicted for diarrhea separately, dyspnea separately, nausea separately, pneumonia separately, pyrexia, which is fever, and vomiting separately. So, and then I looked at the scores. F1 score here is how well the model is, or how accurate the model is predicted. It is not the same as the accuracy score. So the accuracy is not the right metric to use in the case of classification models, because it can be accurate. It only takes into account the true positive and the false positive, but it doesn't take into account the false negative and false positive. So F1 score actually takes into account both sensitivity and specificity. That is, it takes into account both true positive, false positive, true negative, and the false negative. So it's actually a harmonic mean of sensitivity and specificity. So when I did the F1, when I looked at the F1 score, none of these models really did a very great job. It was about 60 to 70% was the F1 score. So the higher the F1 score, the better is the model. That's so if you get like 85% or 90% as the F1 score, then the model is really good. It's accurate. That's what it means. In addition, F1 score alone may not be enough for as a metric to predict, to see if the model is good or not. So I used the AUC ROC score. Basically what the AUC ROC score does is it sees how well the classification model is performing. So basically higher the score, it is a probability of the model. Basically. So it is between zero, the values between 0 and 1, and the higher the score, the better is the model. So when I looked at the AUC ROC score percentage, again, for each of those side effects, it was not really high. It was the probability is only like 55%. Confusion matrix is another metric that I used. So this one actually looks in detail of how much of true positive and how much of true negative the model predicts. And also how much of false positive and false negative the model predicts. So for example, in case of diarrhea, it could predict 9,652 drug pairs having a specific side effect, which is diarrhea. But it also predicted falsely 2,116. And also it predicted false 5,727. So basically the higher the true positive and the higher the true negative, and the less the false positive and less the false negative, the better will be the model. So all these models again predicted overall approximately the same amount of true positive and true negative. And there was a high amount of false positive and also high amount of false negative. So again, this probably is because I selected for the most common side effects for the drug-drug pair. So what I learned in this step, in this whole process, is that this is a huge data set, which I mentioned is a long as well as wide data set. It required multiple merges, and I had to confirm at every step that the drug-drug pairs profiles along with the chemical substructure profile is not mixed up. So that's very important to make sure before we do the model. And since it involved multiple merges, I had to confirm at every step. How are we doing the time? Okay. And since it required a lot of computing power, I had to not only reduce the size of the final data set, I also had to take into account several assumptions and also simplify before I ran the model. As I mentioned, there are several limitations of the model. Here, I didn't specifically calculate the F1 score or AUC ROC score for all the side effects for each of the drug-drug pair. I did it for each of the side effects. So that is an oversimplification. And I used only like 6 most common side effects for this model building. And the drug-drug pairs so the side effects can, as I mentioned, can be caused not only due to chemical structure, it can also be caused due to the interaction of the drugs with the proteins and genes in the human body. But in this case, I only used a chemical substructure profile of those drugs. What I could do with this data set in future is that maybe I can look at the least common side effects and build a model and see how the model predicts. Or I can do some, I can maybe look at better prediction model like multi-class or multi-label prediction model to see if that helps. As I mentioned, the prediction of side effects is due to interaction of drugs with other chemical compounds in the human body, like genes, proteins, or enzymes. So that's something else that I can add and see how the model predicts. So just to conclude, as I mentioned, the data was imbalanced, possibly due to the most common side effects that I selected for the model building. And the models performed only moderately well to predict these side effects. And they were high false positive and high false negative in this models that I ran. Should I stop here for questions before I show the link to the GitHub and just walk through some of the notebooks?
Sure. Does anyone have any questions?
This is LeVar. If you're going to continue on, well, excellent presentation, by the way. Heck of a data set. If you're going to continue on expanding your research and looking for the interactions, I'd suggest looking at some medications have effects where they enhance the effectiveness of other medications. They reduce the effectiveness of other medications. They increase toxicity when worked in combination. So there's, in addition to just like the purely the side effects, look at the interactions of the medications themselves and the organ systems like liver, kidney, how they're excreted, etc., when considering how they interact with one another.
Yeah, that's a very good suggestion. Yeah, yes, that's something else I can do. Thank you so much.
If you want to connect afterwards, I can send you some papers on looking at that specifically. And
yeah,
boy,
that'll be definitely great.
Where you've made it so far, I mean, I don't want to sound critical, but you've made it such a long way. I think adding this type of thing would just be a small step to what you've already done.
Thank you. Definitely, I will. I'll keep in touch with you to get those papers.
All right, let's hop into it.
Okay, I have to stop here. And, all right, so this is the link to the notebook for model building and for data exploration. So I don't want to go into each of these notebooks and go through each step, but I just want to let you know that each of these are numbered in such a way that this is the first notebook. If you are interested, you would want to go and look. So this is where I imported the data set. So I don't know if it will take time. Initially, when I started this project, I didn't know which data set to use. There were several data sets, but so there are several data sets, but those were not like the drug pair. For instance, this one had only like the UMLSID, and then I'll have to go, if I use this, I'll have to go and look what that ID mentioned and it mean. And then I also know if that is a drug pair. It's not a drug pair. So finally, I ended up with using those two sites, which I mentioned. So, but if you're interested in looking into any of these data sets, these are the different data sets that I explored initially before I settled in the two sites and the chemical substructure profile. And then once I decided on the data set, I wanted to merge that data set with the two data sets. So I have the two data sets, which is called two sites and the chemical substructure. The two sites contained the drug pair and the corresponding side effects, and the chemical substructure had the chemical substructure profile of each of those drugs. And I had to merge this and do all this pre-processing, which I have done here. And then this is where, and then there's a little bit of data exploration, and this is where I start building the models. So each of those notebooks will be separate models. So I just initially started with logistic regression without any oversampling. And then when I ran the model, I felt there's something wrong. So I had to go back and look and see what caused that, what caused even though I did balanced class weight, I still didn't get a very good score. So I went back and looked and saw that it's an imbalanced data set. So I wanted to oversample, use oversampler to balance the data set. So that is what is called as logistic regression with oversampling. And after that, I did everything with oversampling. So then there's random forest, XGBoost, and then the lasso regression. And so I guess that's all I have. And each of these notebooks will show how I divided the data set into test data set, and I also had a holdout data set. And also it'll show the confusion matrix, what is precision and recall. So this is where I said F1 score is a harmonic mean of precision and recall. So precision in other words is called specificity, and recall is otherwise called as sensitivity. So how sensitive the model is in finding whether the side effect is present or absent. And the F1 score, it'll also show the F1 score. And I also calculate the accuracy score initially, and then I felt that was not that alone will not be a good metric. So I ran ROCAEC score and also F1 score, and for each of those side effects. So everything is here in the notebook. If you have any questions later on, please feel free to contact me, and we can discuss and learn from each other.
Also, I will be sharing that amongst all the other links. So you'll be getting a message from me in the next few days that I'll include a link to the GitHub, the recorded video and such.
Any other questions?
I'm just curious. So it could have been interesting to also, because you wanted to look at the drug pairs, right? And but you didn't filter it down by just drug side effect interactions.
Drug side effect interaction? No. Yeah, go ahead.
So I was just thinking that, I mean, it's a great, maybe that would have made it the data a little bit cleaner, because especially if you're using common side effects, right? Because some of those could be caused by a single medication.
Yes, specific. Yes. That's true. So yes, this is just the first step towards model building and seeing what I get. And this is always an ongoing project. Yes, I have not used interaction between the side effects. I only looked at the interaction between the chemical structure profile between two drugs to see specific side effect.
Any other questions or comments?
I would just like to say how amazing of a job you've done with this, Suneethi. So thank you so much for sharing this awesome information. I know personally I've learned, and every time I join this conversation, I always learn something. You did awesome presenting the information, very clear, very direct. And you also simplified it for us that are not as technical as others. So I'm grateful for that. Thank you.
Thank you. Thank you. Happy to hear that. Thank you so much.
Awesome. Well, thank you, Suneethi, for bringing this topic, and thank you everyone for coming out. As I mentioned, I'll be putting together a group of links and sending that in a few days. And yeah, thank you everyone. Hope you have a good night.